Okay guys, so this is a very short video. I'm not going to waste time. I'm going to straight go into the mechanism. So we are going to start the mechanism. Like basically we are going to talk about the biosynthesis of Coumarin. I'm going to start our synthesis with cinnamic acid. If you are wondering like how we can synthesize the cinnamic acid, we have talked about in our previous videos. I will put the link above in the right corner. You can go back and refresh your memory if you want. Otherwise you can continue with this. So basically what we are going to do is, but not we are going to do in biological systems, first step is the aromatic hydroxylation. Here you can see OH2 piece attached to this one. Now here this is a very important point. Let me rephrase it. If you remember in our previous videos, like this cinnamic acid, the same thing we have done with aromatic hydroxylation for the same cinnamic acid, but instead of attaching hydroxyl group into this position, in our previous video we have talked about that the hydroxyl has attached to this position, right? So like you may wonder like how we can position this particular hydroxyl group in a specific position for this benzene group, right? As I have told you before, like these are enzyme driven reactions. So enzymes are doing all the parts. Enzymes are the compounds which are deciding whether, when, or which place the hydroxyl is going to be attached. That's why in this case we have hydroxyl on this place, right? So don't get confused. These are enzyme driven reactions. So if the enzyme decided to place it here, so it's not the Coumarin biosynthesis, right? It's not the Coumarin biosynthesis path. It's the other path. So in this Coumarin biosynthesis path, we are going the enzymes are going to place the hydroxyl group in this particular place. Okay, so let's move forward. So let me erase this one. Okay, so like this is a trans, right? Here you have a hydrogen, here you have hydrogen. This is a trans structure. So these are two isomers, stereoisomers. So basically these are not these are superimposable. Like I hope you know about the isomers. These cannot be rotated, right? This molecule can rotate this bond to form this one. No, that's not what I mean here. The sample contains both of these structures. That's why I have put here. But instead of that, I would let's put, yeah, I cannot guide it. If I write a plus sign, it would be more meaningful. But it's okay. You got the idea, right? So this is two molecules, right? These are two molecules, this one and this one. So our enzyme will be driven from this particular structure, this particular orientation. So basic esterification. So these things are normal organic reactions. I hope you know about that. So this hydrogen is going away with one of the hydroxyl groups to form a water. So water is going to be removed to form the esterified coumarin structure. So this is our final target molecule. Pretty easy one, pretty simple biosynthesis method. I hope you got the idea about it. In our next video, let's move into another part.